Hyperbolic, sigmoidal, and an all or none response. The relationship of signaling molecule concentration to the intensity of the response can be either hyperbolic, where the intensity of the response is proportional to the concentration of the signaling molecule, sigmoidal, where the intensity of the response increases much more at a higher concentrations of signaling molecule, all or none, where there's little to no response until a threshold concentration of signaling molecule is reached, then there is a full response. Examples of an all or none response include cell division, proliferation, differentiation, apoptosis. An all or none response is an extreme version of a sigmoidal response. The following molecular mechanisms are responsible for making a response sigmoidal or all or none. Positive feedback loops in the pathway. For example, this simple pathway with no positive feedback loops would be hyperbolic. Whereas this signaling pathway, where there's a single positive feedback loop, the signaling molecule concentration to response will start to look sigmoidal. In our third example, we have two positive feedback loops, and this will start to make the response more sigmoidal and push it toward an all or none response. The relationship between signal molecule concentration and response will move towards an all or none response the more positive feedback loops there are in the pathway. The requirement of multiple upstream molecules binding to the immediate downstream molecule in order to activate it. Example. Imagine we have two molecules. The blue molecule can be only activated if the red circle, a separate molecule, has occupied 6 binding sites on the blue molecule. And we have the following pathway. Signal molecule activates the receptor activates protein A will activate the circle, and the circle will go on to activate our blue molecule, and then the blue molecule activates protein B, which is the effector protein, which then goes on to activate the response. Now imagine the following scenario. There are 5 copies of the blue molecule, and the concentration of the red molecule is slowly increased. At low concentrations of the red molecule, you will have some binding spots in the blue molecules occupied, but none of the molecules are fully occupied, so none have been activated. At medium concentrations of the red molecule, you have more of the sites occupied, but again, none of the individual molecules, the blue molecules, have been fully occupied, so none of the blue has been activated. At high concentrations of red, again, you run into the same problem. You have one molecule all the way on the left that is almost fully occupied, one of the blue molecules, and the rest are still missing a few red molecules to become fully activated. So none of the blue have been activated, even though the concentration of the red has increased. Remember, the concentration of red increases because the signaling molecule is activating the receptor. At very high concentrations of red, now you have a couple of the blue molecules fully occupied by the red molecule and activated, whereas the other 3 have not been. So 2 of them have been activated. So this makes the signal molecule concentration to response sigmoidal, because at low concentrations you're not going to have any response, and slowly when the concentrations start to increase, you start to get a response because the empty binding spots start to get occupied. Example, activation of pKa through ACAP inactivation. And again, here in this example, the ACAP, the regulatory subunits of pKa, which are bound to the kinase subunits, keeping them inactive, you need a total of 4 cyclic AMP to bind to those ACAP subunits that activate pKa. Now we have to remember, we have a picture of a single molecule here, but within the cell, there are many copies of this molecule, so many binding sites, and there is no rule that once one cyclic AMP binds to one of the binding sites on a single molecule, that the other 3 must be occupied. It is completely random and probabilistic. Thus, at low concentration of cyclic AMP, you're not going to get any activation of pKa, but as the concentration of cyclic AMP increases, then you're going to have some pKa molecules, they're ACAP subunits that are bound to them, keeping them inactive. Those ACAP subunits will start to get fully occupied, therefore activating pKa. So anytime you see some sort of requirement such as this within a pathway, you know that pathway is not hyperbolic, that it's going to be sigmoidal. Requirements that a protein and pathway must be phosphorylated at multiple sites in order to be activated. Same principles as 2, but instead of binding to multiple sites have to be phosphorylated. This is the same requirement as the one we had previously. For example, you have a kinase activated upstream, and that kinase must phosphorylate its target. But for the target to be activated, it must be phosphorylated at maybe 2 or 3 different sites, or 4. Each different site is a single substrate for that kinase. And there is no rule that once one site is phosphorylated in a single molecule, all the sites have to be phosphorylated. So you run into the same issue. So if you see within a pathway that a molecule to become fully activated requires multiple phosphorylation events to occur, you know that that pathway, the signaling molecule to response, is going to be sigmoidal and not hyperbolic. A single occurrence on one of these mechanisms will lead to the relationship of signal to response becoming sigmoidal. Multiple occurrences of the above mechanism in a pathway would lead to an all or none response. Cell cycle R point, an example of positive feedback leading to an all or none response. Cell division is an all or none response. Cell cycle progression is controlled by cyclin proteins. For a cell to get past the R point, it must activate cyclin E. Cyclin E is activated by cyclin D. Cyclin D activation is dependent on the activation of growth-promoting pathways such as the MAP kinase pathway. RAS activates RAF. RAF activates MEK. MEK activates ERK1 or 2. ERK1 or 2 will activate AP1. AP1 will activate cyclin D, and cyclin D will activate cyclin E. And cyclin E will push the cell past the R point. The R point exists in this activation step. Cyclin D activating cyclin E. Cyclin E is inhibited by P27-KIP1, so we're showing that inhibition step on cyclin E below. Activation of cyclin D leads to the activation of existing cyclin E pools in the cell. So cyclin E levels are pretty low in the cell at this moment, prior to activation of cyclin D, and cyclin D will go on and activate the existing pool of cyclin E within the cell. Cyclin E activates its own expression. This is a positive feedback loop. By expression, we mean gene expression, so more cyclin E is made. Cyclin E will inactivate P27-KIP1 by phosphorylation, thus removing the inhibitory effect of this protein on itself. This is a positive feedback loop. So you have cyclin E inhibiting its own inhibitor. That is a positive step. The combination of these two self-enforcing feedback loops and other mechanisms not mentioned lead to an all or none response, and this is the R point. Once activated cyclin E levels increase, then cyclin E will go on to activate cyclin A will go on to activate cyclin B, and this will push the cell through the cell cycle into the S phase, G2, and M phase, where the two daughter cells will be made.